The overall goal of these three complementary experimental techniques, namely yeast-2 hybrid, co-immunoprecipitation, and chemical cross-linking, is to assess protein-protein interactions, and in particular self-association, and to determine the stoichiometric composition of protein homo-oligomers.
These methods can help answer key questions in the field of channelopathies, such as altered Ryanodine Receptor oligomerization underlying cardiac arrhythmias and sudden death. The main advantages of these techniques are their relatively high throughput screening, ease of use, and highly reproducible results, while requiring minimal equipment and reagents.
The implications of these techniques extend toward therapy of arrhythmogenic cardiac disease because they help establish the molecular mechanism of action of antiarrhythmic drugs.
Generally, individuals new to co-immunoprecipitation will struggle because of potentially discarding the protein agarose beads, which are very small, when aspirating the supernatant during the centrifugation steps.
In this yeast-2 hybrid assay, yeast cells in mid-log phase are used for transformation. Harvest the cells by centrifugation. Resuspend each pellet in 5 milliliters of sterile water and pool the cell suspensions. Centrifuge the cell suspension. After discarding the supernatant, resuspend the yeast pellet in 1 milliliter of freshly prepared sterile 1,X lithium acetate and TE. The competent yeast cells must be used within 1 hour of preparation.